Welcome back, everyone, to the fifth talk in my science series on epigenetics. In order to better understand epigenetics and how we can affect our genes, it's very helpful to have some knowledge of what our DNA actually is and what role it plays in our body. Once we understand our DNA and how it works, then we can start to look into all of the ways gene expression is controlled. Gene expression, or how our genes are used, is the key to epigenetics and our key to being in charge of our empowerment. This is a review article published recently, February 2024, in the prestigious journal Nature. Genes are not the blueprint for life. The view of biology often presented to the public is oversimplified and out of date. Written by Dr. Dennis Noble, professor of cardiovascular physiology at Oxford University. He goes on to summarize in his own words, "Standard biology has got causation upside down. Genes do not create us, body and mind. We control them and change them when we need to." These potent statements underscore the power of epigenetics. Epigenetics is the science of how our lifestyle and environment can influence how our genes are expressed without changing the underlying DNA sequence. We need to move away from the gene-centric approach that our DNA is completely in charge, because it is simply not true. As we've discussed in my earlier talks, the DNA stores a lot of information, but it does not decide how that information is used. Our perception of our environment, our lifestyle, and our beliefs determine how we choose our genes. You may ask, why is it important to learn about the function of our DNA and the structure of our DNA? Well, it's a very interesting molecule to learn about, but beyond that, we need to understand the control mechanisms of our DNA if we want to affect those mechanisms. My name is Robert Needleman. I'm a molecular biologist and biochemist. Thank you for joining me for a few minutes. Welcome to Empower My DNA. Here's an example. Have you heard of the Library of Congress? It has 3 main buildings and houses over 178.2 million items in its collections in 470 languages. Representing our DNA in this analogy, the library holds a large amount of information. However, it takes us to go into the main reading room to access the information we choose to use. The library's information is only used if we access it. The same is true for our DNA. Our DNA has on and off mechanisms, but they are not controlled by the DNA. In order to understand how to affect our genes, we need to explore how our DNA works. When we look in the mirror, we see ourselves. But our body is actually a community of about 30 trillion human cells and 39 trillion microbes in our microbiome. To better understand how to influence our epigenetics, we need to explore how our cells function. The definition of epigenetics raises several questions. What is a gene? What is gene expression? What is the DNA sequence? Okay, let's dive in. What does our DNA do? Our DNA stores information, whether you think of it as a solid-state drive in a computer, or an encyclopedia of books, or a computer server. Our DNA stores information. And what information is stored in our DNA? One type of information our DNA stores is the code to make all of the proteins in our body. Proteins are the building blocks of life. Most of the structural and functional components in our bodies are proteins. Proteins are so important, a gene was originally defined as a sequence of DNA that codes for a protein. This leads to our next question: what is the structure of our DNA? Many great scientists, and especially James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin, made contributions that culminated in the discovery of the structure of DNA in 1953. Watson and Crick published in Nature Molecular Structure of Nucleic Acids, a structure for deoxyribose nucleic acid. This earned 3 of the scientists the Nobel Prize in 1962. Unfortunately, Dr. Franklin passed away at a very young age. So the molecule is named deoxyribonucleic acid, or DNA. Here is the structure of DNA. It's like 2 strands of beads run in parallel. The beads are called the sugar phosphate backbone. In between the strands of DNA are letters called nucleotides or bases. The letters are C, G, A, and T. C always pairs with G, and A always pairs with T. Now, we're going to see the 2 strands extended with the base pairs connecting them on the inside. And when the strands rotate around each other, they form a double helix, because each strand forms a helix. Here is an animation of the DNA double helix from the DNA Learning Center. The magnification is 100 million times. Here we see the DNA unwinding, and you can now see the 2 parallel strands. The strands run in opposite directions. When the DNA is unzipped, you can see the 2 linear sequences of the letters A, T, C, and G. You can see how A always pairs with T and C always pairs with G. One strand is a complementary image of the other. So if you know the sequence of one strand, you can work out the sequence of the other. And this will become important as we learn more about the DNA. Now we see DNA zipping back together and finally twisting into the beautiful double helix. The sequence of the letters A, T, C, and G will determine the genetic code, but we'll save that for another talk. I hope you find DNA as exciting as I do, and what's truly powerful is that the information stored in our DNA is used in any way we choose. The co-creators of Neurolinguistic Programming, or NLP, Richard Bandler and John Grinder, asserted that if one person can perform a specific task, anyone can learn and replicate it. It's a very interesting way of thinking about the fact that anything we choose to do in our life, we can do. We can say things like, anything I focus on and work for, I will achieve. Thank you very much for joining me. We have a lot more empowering epigenetic science to discuss in my next videos, so please join me. Have a most incredible day.